Good afternoon and welcome to a very special edition of Autism Live. We're here to do a very specific webinar about a metabolomic study. That's a mouthful for me, I gotta say. And if it's a mouthful for you guys watching, you're thinking, I have no idea what that is. Welcome to this really informative hour. We're here right now with Torben Kimhofer and Dr. Anisha, and I'm gonna have you say your last name so that I don't murder it.
It's Wijeyesekera.
You see what I'm talking about now, guys, right? That's why I had to have her say it. They're coming to us from Imperial College in England, and I'm going to have you just briefly say what your titles are because they're long and varied, and I don't want to murder that either. So do you want to start, Dr. Anisha?
Sure. So I'm Anisha Wijeyesekera. I'm a research manager at Imperial College London, and my background is in metabolic phenotyping.
And you may not know what that is right now, but I guarantee you before we're done, you're going to know and you're going to be so excited it's going to make you jump around your living room. Okay, and Torben Kimhofer, tell us a little bit about yourself.
Yes, hi, I'm Torben. I am a research scientist at Imperial College London with a background in bioinformatics.
All right, that's a mouthful. So we're going to geek out a very little bit and talk about some really important scientific things that are going on and a specific study that is happening right now in conjunction with Imperial College and with CARD, and we're going to talk a little bit about that. We're going to allow you guys to educate us, expand our minds a little bit about some things that are going on in our bodies and that are going on in our children's bodies. We're gonna gain knowledge about that so that we can understand why this study is so pivotal, why this study is so important. I guarantee you it's going to blow your mind when you hear all these things. And then we're going to talk about how you might participate in the study with your child or with your children and what that has the potential to do and show not only the world, but very specifically the autism community and how it can help us all to get so much further, so much quicker. And that gives me goosebumps, you guys. Thrilled to be here with you. I want to start by talking a little bit about what the name of the study is. We're going to be from time to time referring to some slides because this information is so dense. That's why we're calling it a webinar edition of Autism Live. We don't do that very often, but it's so important that we get this right, that we're going to get a little heady here. But not too heady. I keep saying it's the Metabolomic study. I'm calling it the poo study. Because you heard me if you joined us earlier, we're going to talk a little bit about poo. There's going to be some talk about urine. So I call it the poo and the pee show. But it has the ability to change how we look at autism and how we treat autism. So everybody give a fist bump. Okay, so name of the study, first of all, Torben?
It's a Metabonomic and Microbiomic Phenotyping in Autism. At least that's the title of our talk.
Okay, and we're going to make you say that 3 times fast later on to see if it's a tongue twister too. But okay, and we have to talk a little bit about who our lead researchers are a little bit. This is like a who's who kind of. Somebody very near and dear to my heart, Dr. Doreen Grampiche. And Professor Jeremy Nicholson, who is the head of the Department of Surgery and Cancer at Imperial College in London.
That's right.
It's pretty amazing.
This is a very exciting partnership between Imperial College and CARD. Professor Nicholson pioneered metabolic phenotyping almost 30 years ago and has been very excited to try and apply this to better understand autism. So this is a fantastic opportunity.
It is a fantastic partnership. A lot being accomplished, which is going to be revealed as we go along. But we have to talk about these two. And there's been some funding. We want to talk a little bit about some of your funding sources that we have to say thank you as well.
That's right. We'd just like to say we're truly grateful to the funders of this research project, the Christian Gerhard Jepsen Foundation, without whom this project would not have been possible.
Amazing. Thank you to them. Thank you so much. This is pivotal. Hearing about this as an autism parent has been so exciting to me, and I'm so glad that we're going to be able to share this with other parents as well today. Okay, so Torben, let's jump in here.
Let's jump in.
Educate us.
Okay.
Bring us home.
So actually, Anisha and I would start our talk with a bit of a controversial statement, and that is humans are a superorganism, right? So there has actually been a recent shift in how we perceive as humans in health and disease, and not so much as an isolated individual, but an individual embedded in a bigger environment. And that environment particularly includes a microorganism, right? Bugs are surrounding us and live on us and within us. And there was a shift from seeing bugs more as germs that make us sick towards actually we evolved with all these bugs, right? We are living with them, towards a perception now that is actually we partly rely on this on these microorganisms for our long-term survival.
Absolutely. And for our autism parents watching, you know, you're gonna hear a lot of things said in this talk. They're gonna excite you because there are things that autism parents have been talking about, that our kids are living, breathing organisms,
Right.
and that they have things going on in them, and welcome home.
Yes.
That's what we're talking about.
Absolutely.
Yes,
Yeah.
that might be a bit of an awkward thought for many people, that we have actually microbes everywhere and we're trying to make it microbe free all the time, but actually we're not sure if that is a good idea.
They're not necessarily only a bad thing. They can be
Exactly.
good for us.
Right.
And that's so important to understand that there are good bugs as well as bad bugs.
Yes.
Exactly. So we particularly care about the bugs inside our gut. The bugs inside our gut make up approximately 1 kilogram of our total weight.
Wow.
Right. And we have approximately, I think that was tens of trillion bugs in our gut. That how much is a trillion? I think that is a 1 with 12 zeros at the end. So that is more than the world population. A human population on the world. So
Amazing.
yes,
It's kind of like Horton Hears a Who going on in our gut. There's a whole village in there.
yes, that is not only a village, but it's all micro universe in our
A
gut,
whole
right?
system there that's dependent upon a whole bunch of things in
Exactly.
order to function properly.
Exactly.
Okay.
So though these are a lot of microbes, there are approximately 500 to 1,000 different species, or different types of microorganisms in the gut. And the composition, so which species are present and which not, they depend on a variety of things, such as the mode of delivery. So children who are born vaginal have a different microbiota than children who are born with a cesarean, for example.
Really? Okay,
Yes.
now that's fascinating already.
Yes.
Okay.
So there are a couple of things that influence our microbiota in the gut. For example, also the diet, right? Which drugs we take, antibiotics, we all know that kills the germ. If they are killed, they have to be re-established.
Right.
So this all influences our microbiota, and many other things as well. Maybe a bit more controversial is if you have a long-term partner and who's your long-term partner, or if you have a dog or a cat, that all influences which microbes we cultivate in our gut.
Interesting.
Right. So we are looking at at these guts from a different perspective. How important are these guts in our health and disease? So we know that certain diseases are associated with a shift of this of this microbial composition in our gut, and that actually has been associated with a couple of diseases.
Okay. So, and that's of particular interest to you guys, wanting to know exactly, looking at a picture to see. We're a little bit ahead slide-wise, I think, than of ourselves, but that's
there
okay.
It is, yeah, sorry. That is actually a particular example that is has emerged in recent years. And
Let's go ahead and full screen that so that we can all look at that together.
That has emerged in recent years, the concept of the gut-brain axis. So we know that certain microbiota in the gut, they produce certain chemicals, or call that microbial co-metabolites, that are uptaken by our gut and travel through our system. Now these chemicals, they have a lot of influence on our body, and that they activate certain signaling pathways in our brain. And these signaling pathways have been associated with the level of stress, or how we cope with stress, anxiety problems, or mood behaviors.
Okay, so this, for some people, this is still controversial and the jury is still out for them, but science has shown us that there is a gut-brain connection.
Yes.
Every single autism parent knows that there's a gut-brain connection.
Okay.
You're preaching to the choir here. We're like, go Torben, because we see this and we know this, but a lot of times we have trouble convincing our pediatricians that this is a thing, that there's a gut-brain connection. But you guys, you have the science for it. You know that there is.
Yes.
Okay. Yay, Torben.
So it might be not of a big gap then, if we think about mood disorders or behavior that we think that we associate that might be with ASD. autism spectrum disorder. So we know that people on the spectrum, or many people, individuals on the spectrum, exhibit a certain behavioral pattern.
Yes.
And that might also be related to the gut microbiome, right? So do we have evidence for, or what evidence do we actually have for this? And I think there are a couple of points. Yeah, the slides you should see in full screen. We know, we talked to CARP, but also other organizations and clinicians, and they all report that concerned parents and caregivers often report distress in people on the spectrum, right? Other evidence, we know for certain research studies suggest an association between GI distress, sleeping problems, and ASD, right?
Absolutely.
So there are a couple of really clear hints towards that. However, there are also unequivocal reports on the association between severity of gut-type dysbiosis and autism symptom severity, right? So it's not everything is clear. There still has to be research done.
And we were talking earlier about there's going to be some jargon terms that are going to come up, and I'll stop you whenever we get to them. So we're going to stop at dysbiosis.
Right.
New one for me. Tell us what dysbiosis means.
Yes. So dysbiosis basically means that the bacteria we got are in a different ratio, different compared to the healthy individuals, right?
Okay.
So we know that certain shift in gut microbiota, there's a ratio between this bacterium and this bacterium that has changed in dysbiosis.
Okay, so it's different. So whenever we come to dysbiosis, we're going to think different.
Yes.
Yeah.
Got it.
All right.
Okay, moving on.
Moving on. Yeah, so there has been a mouse model developed for ASD. And there is one study that clearly shows that if these mice are given a certain bacterial species, prebiotic, then this improves skills related to communication, sensomotor, or anxiety behaviors.
Which we've been talking a lot on Autism-Live about that particular study and how interesting that mouse model, but it's interesting enough that it has our interest peaked, right?
Exactly. So obviously that mouse model can't be easily transferred to humans because it's but a very different system.
Okay.
Okay, even in humans there are cases where a change in diet can significantly alter the behavioral pattern. However, that is not always the case per se. There is a lot of research which still has to be done. However, we think that this is some evidence which should be followed up.
And I think it's fascinating because we hear people all the time that will talk about this diet or that diet and what results they got with their kiddo or what results they didn't. And when people do enter into an autism diet, they want that big bang, you know, they want that big result that they've heard from some people, and some people get it, some people don't. And it's been one of the problems that we haven't been able to figure out why with some kids there is a change and other ones there aren't. But part of what you're doing is going to shed some light on that.
It's going to address that, because what's so important to realize is that every child is an individual. So what might work for one may not work for another. So the ultimate goal of this study is help to support personalized healthcare and, you know, identify the optimal treatment strategy for every child as an individual, not just as a blanket. This will work, this won't work.
Very exciting. But we're still educating ourselves about this idea that there are these microbes and they play a role, we think, in autism.
We think that, and that is basically what our study is about. We'll come to that later again.
Yes.
So I hope it's not getting too technical right now. I just want to tell you how we're planning to investigate that relationship.
Absolutely.
Right.
Is this where we start to talk about the poo?
This is where we start to talk about the urine first.
Okay, we're going to talk urine first. I'm sorry, I rushed things.
Don't rush things.
Talking about urine. Okay.
So actually, it might seem to all of us a bit, or when we started, a bit awkward actually, that we're looking at something that we rather flush down the toilet and not be bothered again. However, we have experienced that actually urine and poo are quite informative for us from a Metabonomics perspective. I come to that, what that is. So in urine is basically everything excreted from our body that we don't need anymore in terms of metabolism, right? And by looking at these chemicals in the urine, we can work out what actually is going on in our metabolism in our body, and not only of our own metabolism, but also the metabolism from the bacteria in our gut. Right, on this slide, I don't know, you see that now in full screen. We see a urine sample and a spectra right next to it. And this is the spectra we're actually looking at. So each peak gives rise, each peak is a signal coming from from one compound in the urine, and that we can not only see our own metabolism in there, but also the dietary components. As I said, I can work out what your last meal was if you would give me a urine sample now.
And if you had a glass of wine
if
with
you
your
had
dinner,
a glass
we
of
can
wine as
see
well,
that.
yeah. So
Right.
these are very sensitive methods, right? With not only dietary components but also microbial co-metabolites, so the chemicals that have been produced by the microbes in your gut and that have been uptaken by your body. We can see all that from only one urine sample.
It's amazing.
Yes, absolutely. So maybe formulated a bit different, it gives us a snapshot of what is going on in your body right now.
Wonderful. And just from the urine. And I know we're going to talk a little bit later on about how you get a urine sample from a child who's on the autism spectrum, because that might seem overwhelming, but it's a heck of a lot easier to get a urine sample than to get spinal tap fluid
Exactly.
or
a
blood
sample.
or anything else. You know that your child is going to urinate at some point during the day, and you can catch it. We're going to show you how to. So great, because we get a bigger picture of our kids, our total kids.
Absolutely.
And that's just the pee part. Now we're going to talk about the poo.
Not
I'm
really.
still rushing
No,
things.
I know
It's
you're
like I
very
want to talk about the poo, Torben.
No, you're very excited
all right. I will
about that.
calm
I'll
myself
promise
down
you-
about the poo.
I'll
I said
promise
this
you
was
we
the
spent
poo
a
show.
lot of time with you.
Okay. All right.
Don't worry.
I said when the poo hits the fan. It doesn't work if I say the pee hits the fan.
Well-
Okay, so still talking about the pee.
Yes, talking about P, or the method in general, that is actually called Metabonomics. I don't want to be too technical here. I'm a researcher, so I sometimes get carried away. Please stop me.
I will.
Just give you one definition what Metabonomics is. The first part here is behind Anisha now.
And we're going to full screen it for you.
That's great. So the first part here is a very technical one. I don't want to be too detailed in that. And that was actually coined by the principal investigator of that study, that is Professor Jeremy Nicholson and colleagues, right? And more easy and more amenable definition is maybe the second one. Read it loud maybe. Metabolic
Yes.
profiles capture the complete set of small molecule metabolites present in the system at a given time. That is what I meant with snapshot.
Yes.
Yeah? And this allows us to see drugs, food, microbial co-metabolites present in the body, as well as mapping your own metabolism, the human metabolism.
Wonderful. So it's
Right.
going to get all of that in one urine sample.
Yes.
Powerful.
It's very
And
powerful.
you may think, you may be like, I don't know how powerful. Wait, we're going to show you how powerful. But able to do that with one urine sample.
Yeah,
Spectacular.
exactly.
Okay.
So of course this is implemented in a wider strategy, right? Personalized healthcare is a big issue right now, and picked up by the media as well. It is the way moving forward, and we think we can contribute that. If you think about that, the metabolites, or that what we can measure, is influenced by so many things. So we have the microbes, right? Not only in our gut, but also in other places. And we have our genes, the genetic makeup. They contribute substantially in what we see, what metabolites we see in urine or any biofluid and tissue, but also the proteins that are in our body, and the xenometabolite, that is a bit of technical. These are, for example, what drugs we take.
Or just anything in the environment that might influence your health. So
Exactly.
all of those factors help to shape your health, and that's really important to bear in mind.
And as a parent of a child who's on the autism spectrum, we're always filled with questions about why. Why is my child on the spectrum? How is my child on the spectrum? And how am I going to get my child as healthy as possible? And how am I going to help my child to be everything that they want to be able to be, right? And when we go to doctors, it tends to be that we're looking at things in different slides. You can go now and get genetic testing, and we're all for that, and being able to, because we see that in some cases, especially with kids with Fragile X, boy, that's important, and it's going to affect some of the different things that we do medically with that child. And then you're looking at another slide, and you're looking at what trauma your child might have gone through with their body. And you keep looking at all these different slides, and you're saying you can look at urine and look at the whole picture.
Exactly.
I think that is a general thing at the moment, right? So we're going to different doctors, for example. These are all specialized, but we should be able to combine that data and get a more systems approach. We call that a systems approach, and that is what we do care about.
Wonderful.
So actually, that is here on the slides. It's called global systems biology. The idea is to create a model that considers different types of data and is able to explain the phenotype, what we see, for example, if there is a person with autism.
Okay, but let's talk about phenotype, because for a lot of people that's a word they're not familiar with. So what does phenotype mean?
So a phenotype is basically that what we observe, right? So in this case, we observe a person with autism. Although there might be subgroups, and it's probably important to distinguish between different subgroups. We don't know actually now if there are subgroups. However, phenotype is something that we observe, for example, an individual of spectra. And how do we explain that? We can't explain that purely with genetics. We can't explain that purely with proteomics or the proteins or other things. We need to have a combined approach to explain that phenotype. So what we observe.
So tell me, because when I explain it to people, and maybe I'm taking it too far down, but I always say that, when I think back to, say, 1900, when they talked about cancer, and cancer was all encompassing. It was just, somebody had cancer. And if you went for cancer treatment, they had one thing and people died.
Yeah.
People died left, right, and center. And then as the years went on and they did research and they found out, oh, well, there's lung cancer and there's pancreatic cancer. And it was like, oh, and we treat lung cancer different than we do pancreatic cancer. Now, if people get diagnosed with cancer, they go, oh, I have type 34A, slow growing, small cell, this and this is the protocol to treat this, and people live. And if we can get autism to the point, because I know I meet a kid who's just like my kid and I go, oh, you have the same type of autism that my son has. Yeah. But then I'll meet another kid and I'll meet that parent and I'll go, oh, they have a different type of autism than my child. Doesn't make it good, bad, or indifferent, right? But we all can look at that as intelligent people and go, maybe different things would work with those different kids.
Exactly.
And if we could say, oh, someday, if we could say, oh, your child has type 32B autism, and how we treat this is with 40 hours of ABA, and by the way, there's this probiotic we know to be effective with this, we can get our kids better, faster.
Right.
That's right. So by describing, all the features associated maybe with a particular area, then you're able to do patient stratification, and then that for this group of individuals, this is the best therapeutic strategy.
Yes,
okay.
I completely agree. And also, that is what you just described. Isn't that the concept of personalized healthcare?
Yes.
Right? We know we're all different, and we can classify. Okay, this cancer occurs in the lung. However, we see that the genetics of that cancer are very different, and the optimal strategy would be a different cancer drug, right?
Yes.
That is actually tailored exactly for that genotype. And there we are, another type, phenotype and genotype. Genotype describes the genetic makeup of, for example, a cancer.
Okay, so getting it specific.
Right.
So we can treat it specifically.
Absolutely.
Oh, I get excited. I get really excited about
That's
that.
great,
No,
yeah.
It's really thrilling. Okay. So now you're going to talk a little bit about some of the toys that you have so that you can look at the poo and the pee.
Yeah, so we have different analytical platforms, and they're very sensitive. Let's call them advanced analytical platforms. For example, the NMR, right? NMR means nuclear magnetic resonance spectroscopy, and you can think about that as an MRI, but for liquids or tissues.
So where you guys are hanging out, you're doing MRIs on poo.
Exactly.
So I imagine lunchtime conversation is really interesting.
It gets very graphic.
Yes.
And they love it. I love that you guys love it. That you're
Again, there is so much information in a poo or urine sample, you wouldn't believe it. So it's not a waste product for us at all. It's actually very informative.
Yeah.
Well, and I said this to you earlier, it kind of makes me think about dinosaur poo, where everybody's really into collecting and slicing and looking at dinosaur poo. Some people even shine it and wear it as jewelry. I don't think we're at that point with our kids' poo, but thank heaven. But if there's things to be learned from it, then by all means, let's look at it in as many different ways. And you have machinery that can look at it in every way possible.
Yeah, exactly. So these are very sensitive. We can detect actually the tiniest amount of certain chemicals in different in biofluids, right? These are called NMR, nuclear magnetic resonance spectroscopy, but also mass spectrometry based technologies, and these are even more sensitive. And we apply that, we get actually a spectra. Spectra is, I think what I've showed before, is like these peaks there.
Right.
And we generate so much data with these analytical methods that we can't look at that manually. So what we do, we use computers, powerful computers and powerful statistics. So these are multivariate pattern recognition methods or machine learning. We use all these methods just to work out differences between groups, and these groups could be urine samples from people on the spectra versus urine samples from people not on the spectra.
So again, to take it to the simplest level, you're looking for where things are similar and where things are different and where things cluster differently.
Right.
Because that gives you information about that there's something going on.
Exactly.
It'll take further investigation to figure out exactly what, but when you can identify there's something in a cluster happening, that's when it gets exciting.
And that's clustering based on the molecular level. So this is not based on any of the clinical information. This is just looking at the biology. So how similar is a urine sample? And then does that also translate to, some clinical information? Sometimes it's not
We
the
talk
case.
all the time about diagnosing and how there's an element of, there's a checklist of diagnosing autism. But it's never occurred to anybody that looking at the urine, you might be able to tell whether somebody is on the autism spectrum.
You might be able to tell that, but you might not be able. And I think this is what we are trying to find out.
Right.
Right. The ultimate goal, just to finish with that slide, right?
Yes.
The ultimate goal is to integrate different types of information. That could be information from the chemicals in the urine, plus information about the microbiota, and we get that by analyzing the fecal samples, plus clinical information or behavioral information, just to integrate that all to create one model that is able to explain autism spectrum disorder.
Okay. All right. It's a little heady right now, and it's a little outside of us.
Yeah.
But that gives us the background of this is what you're looking at and how you're looking at it. So let's, I think this is about where we start to translate into what the study's actually doing, right?
Yes.
Yeah.
Exactly.
Okay. All right.
So, just to round that up, right? So what we do, we get samples from study participants, and then we store it here at CARD first until they have a couple of numbers, and then we're going to ship to our labs at Imperial College in London, South Kensington campus, and then we apply our spectroscopic methods, generate data, and analyze that data.
Right.
Right. So actually, we'd like to thank the study participants that have given us samples.
Because already there have been people participating.
Already we've had a fantastic response, and we're really grateful to those who have signed up to our study.
Absolutely.
And we've been so excited, we couldn't wait, and we've actually got started already on analysing some of those samples. So Torben's going to show you some of the results.
I just got to stop for a second and say that, for an autism parent, we talk all the time about how we want things to improve. And a lot of times there's legislation we have to slog through, and you got to go to your school district, and you got to slog through this, and you got to argue. This is an opportunity for families to move the dial right here, right now.
Yeah.
And it just means giving your poo and your pee of your kid, and we're going to talk about how to do that and make it easy. And when you stop and think about, okay, so I collect some of my kids' poo, what does that do? That it gets taken to a card center where it's kept frozen, and then it gets shipped to London, and then they're going to slice, dice, and dissect and MRI it and do all these things to it and study it to the nth degree and come back with some pretty exciting things.
Yeah.
That's pretty exciting.
Right.
That's worthwhile.
And I think what's really important to emphasise at this stage as well is that it's not a diagnostic test. So we're not taking your sample and we're going to screen for a panel of things. The whole idea is that it's for discovery. So you're helping to support the community by identifying what's different in your child compared to another child, and using all of that information will build up a better picture.
And now's a good time to say, thank you so much for saying that. Now's a good time to say too that your child's information is kept very safe. No one ever is able to identify your child with their sample. But at the end of the study, correct me if I'm wrong, you are able to see where your child is clustered, correct?
Exactly. So when we do the analysis, we do it blinded. So we don't have any of the study participants identifying information. We're just given a randomized number. But at the end of the study, what we could identify, whether your child appears in our data, are they very similar to this group of children or not? We would be able to identify that.
I think that's a really worthwhile thing, and the bigger picture of making a difference for what happens in the next decade with autism. If that's not exciting to you, it should be. And we're gonna make sure that you're excited by it, because we're gonna show you some things that are very clear that will get you excited.
Yeah, maybe we start with the stool samples that you were asking.
Are we going to talk about poo now?
Yes, I'm
Okay.
talking about poo, but from a very scientific perspective.
Yes, I'm sorry.
I
I
don't want to
light.
make it too awkward.
No.
But we analyzed a specific part of the DNA, of microbial DNA in poo samples, and that
Okay.
gives us information which species or which types of bacteria are present in the poo, and also in the gut, because that comes from there, right?
Okay.
So we looked at that first. So there is a very specific method. Maybe we can get that full screen.
Yeah.
You see basically a 2D plot with red and blue dots, where each dot represents one individual.
Okay.
So the blue dots are individuals on the spectrum, and the red dots are individuals not on the spectrum.
Okay. And first we got a comment on, it seems very clear that there's a separation there.
Exactly.
So something is going on that's recognizable in the poo, and this is just looking at DNA.
This is looking at the DNA of the microbes. Exactly.
Okay, the DNA of the microbes, not the DNA of the kids,
No.
the DNA of the microbes.
Exactly. The microbes in the
my
poo.
goodness. Okay, the microbes of the poo.
Yeah, so what would that mean? So if you would give me a poo sample from, I don't know.
I don't know you that well,
No.
Torben. I cannot give you a poo sample. No, I'm sorry. All right.
Maybe from your child.
Yes,
He's on-
yes. I know you well
he's
enough
on
to
the
do that.
spectrum,
Yes,
I
yes,
believe.
yes, he is, indeed.
So if I wouldn't know if your child would be on a spectrum
Right.
or not, and would give me that poo sample and we would analyze that, could probably tell you if that kid would be on the spectrum or not.
Because you would look at the DNA of the microbes, and
Yeah.
you could plot where he is on that chart, and you
Right.
could predict whether or not he might have a
Absolutely.
I mean, that already, the fact that there was did you expect there to be such a clear line between?
We were actually surprised that this gives us such a clear signal, right?
Yeah.
However, there are a couple of things we need to consider. For example, what have the children eaten?
Right.
So depending on what you eat, you're going to cultivate different bacteria in your gut. That could be a potential confounder. So if all the individuals on the spectrum have a similar diet, which is different to the individuals not on the spectrum, that could also be responsible for that separation.
Okay.
This is why we would like to emphasize that these are preliminary results,
Okay.
and these have to be validated, which we will do in the future.
But clearly there's reason to look closer.
Absolutely.
Yeah. Yeah, because there's something going on there that we need to know about.
Right.
It's part of that missing puzzle.
Yeah, we
Wonderful.
completely agree. So you might be interested. So that as a researcher, you have the next question you'd ask is why do we see that separation, spectra not on the spectra, right?
Right.
And from our analysis, we can say that this is mostly or mainly because of members of a bacterial genus, bacterial family, Bacteroides. Right, that is a specific type of bacteria that is actually present in all of our guts. However, there are differences. We see observe differences in abundances. So that is a normal commensal of our gut microbiota. However, it appears that certain species are more present in people on the spectra than not on the spectrum.
And specifically, what did you find is more present with the people on the spectrum than it was on? You found one thing in particular, right?
That is called Bacteroides. That is a Latin name for
Okay.
that's bacterial. We call it a genus.
Okay, and so my question to you is, how related is that to yeast?
That is not related to yeast at all, I must say. So
Okay.
yeah, so that is very different.
Okay,
So a yeast comes from it has a different evolutionary path than bacteria. It's still
Okay.
close. A yeast and bacteria are still close, but it's not the same.
because the word that I recognized on your slide was Clostridium.
Clostridium, yes.
And okay, so is that high or that's low in the individuals on the spectrum, or is it different?
That is different, yeah. So we would need to go back, and I wouldn't like to tell you if that's high or low, just
Okay.
because we need to validate that.
Okay, so
first.
I'm jumping way ahead. I'm jumping
Yes,
way
you're
ahead.
very
Okay.
interested, but that's good.
But there's a difference
There
in
is
the,
a
in the
difference
Clostridium.
in that,
Okay,
yes.
all right. I'm fascinated. I just
Great.
want more.
Yeah, you want more.
Okay, all right.
Yeah, so the ultimate goal, not the ultimate goal, but what would be the next step? So we know now that the bacterial composition in autism in individuals on the spectra and not on the spectra is different, and now we would like to link that to our own metabolism.
Okay.
Right? So how does that influence our own metabolism? Is there a difference? And this is going to be my next slide. This is going to be don't want to overcomplicate it that. What we see here is a specific analysis of the spectra that we generated from the urine samples.
Okay.
And this kind of summarizes which signals are higher in people on the spectra versus lower in people not on the spectra. And more important here than the magnitude of the peak is the color. And the color gives us an indication which signals are important for this discrimination between the two groups, right?
And so correct me if I'm wrong, so this graph is showing us these little spikes that go up that are red are things where the individuals who are on the autism spectrum were significantly higher in those things.
Exactly.
And where stuff is in blue, those are things where they were significantly lower and things than the people who were on the autism spectrum.
Absolutely.
So we're
correct.
looking at a picture of what is different metabolically between them and individuals who are not on the spectrum.
Yes, that is correct.
Fascinating.
So
That's the puzzle piece right there, right? I want to understand what that is, and of course that's what you're doing, and we're not going to get to understand all of that today, but the fact that you have a picture of the puzzle is kind of awesome.
Yes. So the next step that we would need to do is look more closer to this and actually see these signals come from these and that metabolites. And then, of course, if we know which chemical compound it is, we might be able to associate that with certain pathways. Metabolic pathways, for example, energy pathways. We all have to generate energy, right? This is a specific pathway, and we know which metabolites or compounds are associated with that. That will be in the next step, and this is actually a very time-consuming step, but we're not at that stage yet. But we do know that there are differences.
Okay, so we've looked at the DNA and the microbes, and now we've looked at what some of the differences are in the urine, but there's a component to autism that we've thought of for a long time, and it, of course, is very near and dear to my heart and to Card's, that's looking at autism from a behavioral standpoint.
Exactly. So that would actually be the gold standard for us, right? So we can connect biological things like what we see in the urine or in the feces or the poo, and we connect that to some behavioral aspects, right? And that will be something completely new. We're not at that stage yet, but we're looking into that because we had also access to the skills database, the CARD skills database, and that gives us information about the behavioral pattern of people on the spectrum, right? So these are some results of that. Again, on the left plot, each point represents an individual on the spectrum, and here colored in red and blue are different clusters. So you see that all the points colored in red are closer to each other, most of them, closer to each other than to the red one, to the blue points, right? So we can observe behavioral patterns or behavioral subgroups based on the skills data, and we would like to link that to actually the urine data, the data we generated from urine and the poo samples. So that would actually, or this study is a unique opportunity to make the link between behavioral patterns and the biology behind that.
And for those of you who are watching who aren't familiar with what Skills is an online curriculum that was developed by CARD. But one of the most amazing things about Skills is that anybody in the world has the ability to get a subscription to it, and you have the teaching tools that my son benefited from, and tens of thousands of kids have benefited from. But there is a portion of Skills that is used for research, that your child is, their information is so coded that it cannot ever go back to the child. But it is helping and has been helping to phenotype autism now for 5 years, Skills, by looking at these clusters, and we're beginning to see, if we teach, this skill and then this skill next, that these particular kids will jump ahead, whereas other kids need this skill in this order, and that's what helps them. So Skills has already been amassing that, and that in this partnership, it already is forming a way for you to take your data and put it with behavioral and make that whole picture become a 3D view.
Yes, I would even say it's not only 3D, but maybe 30D. So
13D. I love it.
It's difficult to imagine that, but it's definitely more than 3D.
My husband's going to want that television set. 13D. I see. Okay. But being able to
make
see
that
it
holistic,
from all,
yes, exactly, and make that picture complete with different data sets. And it might be actually a good idea to emphasize to the audience that every data that we get is completely anonymized. We cannot identify any particular child.
But what it does mean, you guys, is just by doing these things and allowing them to have your data, that this is what needs to happen in order to phenotype autism. It's not going to happen otherwise. This is one place where we as parents get to drive the bus. If enough parents will participate in this and give the poo and the pee samples, and we can all suck it up and do this, then you have enough information to get the data from it and be able to give us back the information that we need.
Exactly, and help to target and personalize therapy.
Yes.
So for example, if we were to use some of the skills data to identify different clusters of kids based on their skills profiles, and then we look at their molecular information and we see a pattern there as well, then if you have another child who comes in and we run their urine sample and we can see, oh, this clusters with that group who actually did well on that particular treatment, then straight away you can have a much faster approach.
Absolutely. This is the good stuff, you guys, and we get to be front row on this as parents. Okay, but we're going to talk about how you can participate eventually.
Yes. Exactly.
Okay. So what are we working towards?
Right, what are we working towards? For us, it's very important to we aim to characterize the different molecular phenotypes of ISD. What does that mean? Phenotype, we explained that briefly before, but basically what we see, we'd like to explain that based on biology, right?
Okay.
So we try this, and I think in this slide it's called endophenotypes. That are even subclusters that we see. We know autism is a spectrum, so we are pretty sure that we can identify certain subclusters, individuals that are more closer to each other than to others outside that clusters, right? I think that is an important task in this, important goal of this study.
We're looking for those similarities and those differences.
Exactly.
Okay.
Also, we want to better understand the relationship between microbial gut composition and host function ISD. So which bacteria or which composition of bacteria influence our behavior, and what functions do they have in our system?
Okay.
Ultimately, we'd like to improve the behavior patterns in people on the spectra through the modulation of the gut microbiota. We don't know if that is possible, but we like to know if that is possible, just to ultimately develop more targeted treatment strategies. And that whole study is designed to basically support earlier diagnosis of ASD.
Okay, so we're hoping from this study, earlier diagnosis, more effective treatment can start if we get it earlier, and to also supercharge treatment with the information that we have so that we give that individual the best possible, most scientifically effective treatment that there is. And streamline that like we've done with so many other areas in the medical field. We've all been crying for this forever, and this study can help to push that conversation and push those ideals further, faster.
Exactly.
Well, you're superheroes. All right. So what are we asking for our folks at home to do? How do they fit into this?
Yeah, so basically, we'd just like to encourage as many people, maybe from this show, or if it's something you've just been thinking about, just do call up to ask for some more information. We're happy to email with more information, or because really what's critical is to have a good number of people participate in the study. Again, as Torben said, it's a spectrum, and we know that people at different stages will have very, very different phenotypes. So what's important to us is to build a very robust picture, that's not based on maybe one or two individuals, but it's based on a good number of people who have similar markers from.
And so let's talk about numbers then, since that's what we're talking about. What are the numbers that you're shooting for this study? What do you need?
So we were shooting for 500 kids on the spectrum.
Okay.
200 siblings, if possible, because again that factors for genetics and environmental background.
Okay.
And then 200 not on the spectrum.
Which, that's 500 individuals who are on the spectrum. This is not a huge number, but it is enough for your study to be significant, to look and see where those patterns and differences are. Exactly. So that's what we need to get, and we all together need to make that happen. Exactly. That is on all of us. If you sit there and think somebody else is going to do it, I got to tell you, what are we waiting for?
Yeah.
I completely agree. I think, it's we ask for a stool and urine sample. That might not be easy for certain people. However, if you think about what you can influence or what the outcome could be, I think it's definitely worth it.
Absolutely.
Yeah, and they were all babies once, and we had to change their diapers, and we got over it. We did it.
Poo happens.
They're a little bit bigger.
And it happens
now.
every day usually. So let's get down to the nitty gritty about this. We have a little poo emoji even.
We do have a poo emoji.
I love it. I'm all about the poo today. I don't know what that's about.
No.
And you brought some kits.
Yeah,
here.
because what I thought,
I've got them on the floor here.
just to kind of dispel that, it's really not technical, it's not complicated, so let's talk about what we're asking you to do. So,
I don't know where my little, you have the urine.
I have the urine, yeah. So
we have some apple juice to simulate.
We have some props to simulate what we're asking you to do.
It's the prop time.
It is prop time.
And you even brought, I was thinking we should have chocolate, but she
we
thought
have
ahead.
Anisha thinks already.
So, so let's start with the stool sample collection.
Yeah, let's start with the stool.
So we understand that, kids might be at different stages. They may still be in diapers. They may be potty training.
Well, we haven't really identified what age group you want. What ages do you want?
So it's going from about 18 months to age 10.
Okay.
And again, I think that's been another source of questions, why that particular age range when we know that it can affect others.
Right.
The main thing with having the age 10 cutoff is, we know as teenagers, you go through different hormonal changes, physiological changes, and what's so important for us in this early discovery stage is just to try and keep it as simple as possible.
Yes.
Even within that age bracket, you're going to have a very different profile between an 18-month-old versus a 10-year-old, and that's why we're hoping that with enough sample, we can actually stratify the data and model them as different age groups.
Okay, so 18 months to 10 years,
Yeah.
which means that there are going to be some kids in diapers.
There are going to be some kids in diapers, so if your child's in a diaper, it's a bit more straightforward. We provide you with one of these stool sample collection containers, which conveniently has a spoon on the lid.
It's got a poo spoon.
It's got a poo spoon, so you just scoop it up,
put
it in from the diaper, close it, label it, put it in the Ziploc bag.
And how much do you need? Do you need just the spoonful? You need 2 spoonfuls? You need a quarter cup?
Ideally, as much as you can.
Okay.
Minimum would be one spoonful.
Okay, great.
But it's all going to waste anyway. So
Right.
if you can collect as much as possible, then that's amazing. None of it is wasteful for us. So we provide 2 collection containers. One, can you see it's got this little clear liquid in it. So what that is that's glycerol. So why this is important is for later on in our study, if we do identify particular microbes of interest, we'll go back to this sample that's been in glycerol because the glycerol will preserve the microbes. So that's why we have a second collection.
Okay, so same diaper sample. So we want them in both
of
things,
them.
not 2 different diapers.
Not 2
same
different
diaper.
diapers, same diaper.
Okay.
And then once it's collected, just put it back in the Ziploc bag in your home freezer. And then when you have your scheduled visit to the clinic, you just take that along with you.
Okay.
And in
but
terms
it needs
of
to
how
go in the freezer.
in the freezer, yes. And in terms of doing it's so easy to use this spoon. So let's do a poo collection, right? So we've got some chocolate here. Right.
If you've all been to when you go to the baby shower and they do the chocolate and the diaper game,
That's
it's
right.
a lot like that. It's just no different.
It's no different at all. I'm sure you'll see caramel in that too.
And if you have to, you put a clothespin on your nose and you get through
That's
it.
right. And there are
We've all
also
had to do this at some point.
gloves provided in the bag
Yes.
as well. If that
And there
grosses
is
you
soap
out.
and water afterwards.
Okay. No, I'm doing a really bad job of this.
No, but you know what? This is the reality of it.
Excuse-
This is the reality of it, and you have to maintain a sense of humor about it.
That's
Absolutely.
right. Absolutely.
Because and then you can blog about it, or you can post on Facebook and say, "I had to collect poo today," and everybody will feel bad for you.
No,
it's not that
Exactly.
hard.
And you can compare, corn and tomato stories.
Oh, now
all
we're
of
going
those
there.
things. Oh, we're going there.
Now we're going
It
there.
gets very
Experience.
graphic.
Right?
It does.
Okay.
So,
Yes. Yeah. So you do that, and
do
you're going
that.
to do it with 2.
That's it.
And you
And
move
so
on.
so this slide just basically summarizes the previous one, the process if you're collecting from a diaper or
Can we
using
full
this.
screen that for them so that they can see the poo catcher?
Yeah, so there's
There's
this
a-
feces catcher as well. So again,
This cracks me up, you guys.
so again, this is it's just a piece of paper which you open up and it sits on the toilet. But again, from feedback from some parents, you know, for some children, this may be very difficult for them to do.
Yeah.
And we recognise that. And, you know, we don't want to cause any additional stress for the child. So, you know, if it's easier for you, you could try putting the plastic wrap on the
I've done
toilet
that.
seat.
I did
And,
that once.
you know, that's also absolutely fine. We want to support you to help you to participate in this study. We don't want to make it unnecessarily difficult. There are certain things, because, you know, as Torben mentioned before, our instruments are very sensitive, so they can pick up on contamination, or particularly from, you know, this PEG. Well, PEG is like a plastic contaminant that's found
So
quite
you'd rather have it on the
frequently,
paper,
We'd
but if
rather
you have
have
to
it
have
on the
it
paper.
on the plastic, you got it. Then you'll do
That's
what you have to
okay.
do. But what
Yeah.
you can't do is let it fall into the
Into the-
toilet and
no.
scoop back out from the toilet.
That's right.
So that's,
Yeah.
you know, and it may take you more than one try to get it. But you got to remember what your purpose in doing this is. It's for a higher purpose. And how often do we get to say that we did something for a higher purpose? And you guys, if this comes back and we get information that makes it easier for one kid and makes that kid have that much more effect, you get to walk tall and know that your poo was a part of that, right? And it's, and you know, it's going to be. But how many times do we have to do it?
So we're asking you to do it once on recruitment.
Okay.
So I think that's within 1 month. We ask you to come back to the clinic with a follow-up sample. So if you'd like to participate in the study for longer, that would be amazing. And if you're able to collect a sample every month, again, that allows us to monitor your child's metabolic phenotype over time, and maybe you might have changed therapy in that time, or you might have started a new diet.
Right.
You know, we'll be able to see what effect that's had on your system.
Okay.
So,
But initially you sign up, you do, when you sign up for the study, you're going to do one, and then a month later you're going to do one more.
Exactly.
And then if you look at it and go, okay, this is infinitely doable, you can sign up to do a longer period of time.
Yes.
But initially you're asking for two poo collections, two pee collections.
Right.
That's it.
Okay.
Yeah.
And I would say, as we, you know, if you are afraid that it's going to be traumatic for your child, the poo collection, you know, first of all, make sure that you're maintaining your composure. Yeah. If you come at it from the point of view of, oh, you know, we're going to poo on the paper and it's going to be fun, your child is less likely to be concerned about it. So keep your emotions all together, and then do something reinforcing afterwards. Congratulate them for pooing on the paper.
Exactly.
You know, it doesn't have to be a big deal, but make it, you know, afterwards congratulate them on doing that.
You know, a bit like when you're potty training, you know, you try to just be positive about it.
Absolutely.
So that will help.
Absolutely. Okay.
Yeah. So that's the poo. Quite straightforward.
The poo did not hit the fan.
Did not hit the fan.
I love it.
You know, urine is even more straightforward. You know, we provide one of these sample collection cups for your child to pee into. So we have some apple juice just to show just how easy it is, what we're asking you to do. So your child will, right, so let's put some fake urine into there. And also in the pack we provide you with one of these disposable pipettes. And she says, so you just open it from the package.
Yeah, opening the package is one of the hardest things. And don't think that we're not going to talk about how you get the pee in the cup. We are going to talk about that. So if you're thinking, oh yeah, she poured the apple juice in there and made it look all easy, but it's not. We're going to go back to that. Once we figure out how to open the pipette.
There you go.
All right, here we go. Okay. So these are disposable. So once you're done with it, it can all go in the trash.
Yeah, don't save it for craft purposes. Throw it away.
Please don't save it.
But they're so fun.
And you know, you just open the cap.
So you do the big collection into the cup because it's a bigger aim.
Exactly.
And you do want to use that cup, not a cup that you have at home. You got to use this
you
cup.
need to drink
That
out
is a
of.
sterile
Please
cup.
use this cup. And you can see on the pipette it has a marker to say where 1 milliliter is. So this is the minimum that we are asking for.
Okay, so you got to have that filled all the
have
way
that
up to
filled.
that
so
top
you squeeze
marker.
it and then you let go and it draws up the urine, and then you put it into the tube and then you press, and it releases it into the tube, and then That's it.
And if you get it wrong the first time, you have more urine from which to dip.
Right.
Exactly.
But you need 3 of those
we
little
need
vials.
3 of those little vials, and again, once you've collected it, back into the Ziploc bag and into your freezer.
Into the freezer, okay, into the
Yeah.
freezer for the urine as well.
As well.
Now, you don't, there's a whole thing around collecting in the cup the urine.
Yeah.
So when you collect the urine, you could just put the cap on it, put it in the refrigerator until the child is preoccupied doing something else,
Yes.
and then do your pipette.
That's absolutely fine.
Because if you're trying to, like, get them dressed and do the
Right.
pipette, you know where you're going to end up with a mess all over the bathroom.
Yeah.
Okay, but let's back up a little bit and talk about how we get urine into a cup. And you aren't the only people that are going to ever ask for urine to be in a cup. You go to the pediatrician and they're going to ask for the urine into the cup.
Yeah.
So it is just, we all need to just go, okay, this is going to happen from time to time. It's not your favorite day, but it's infinitely doable.
Yeah.
By the way, do we have to have the poo and the urine on the same day, or could they be separate days?
They need to be on the same day,
Same
or as
day.
close as possible.
Okay, so you could, say, today's collection day and make
Yeah.
it fun.
Yeah.
But, okay, so some things to, I think, first of all, it's a great thing for target practice for boys. I would suggest that people, if you need to, take them into the shower to get them so that if it goes outside of the cup, you rinse and you're good to go. For girls, you just get that underneath,
Yeah.
right? But you have gloves because you're
You
going to
have
end up
gloves.
with the hand. It's going to happen.
It's going to happen, and I think, as you said, the key is just to make it fun for the, don't stress them out, because chances are they may not just do it. So,
Yeah.
you need to make it fun for them.
And I also want to say, too, that if you feel like you need some help and support, a lot of people that are going to be doing this are skills users, which means that you have a card team and you can ask for some help and support from your team to set it up. You can probably even ask them for some help and support day of. That doesn't mean that, you can't do it on your own, because you can. I just don't want people to be so. This is all that's. This
Yeah.
is all that stands
All that we
between
need.
us
Yeah.
and getting this information.
Yeah.
And we
Exactly.
can't allow a little poo and a little urine to stop us from getting this fabulous information.
That's right, and this is what makes this project so unique, because nobody else is doing this kind of research.
Yeah,
So you have a chance to contribute towards something that's truly amazing.
it is. And you have a reinforcer for the parents for doing it too. So let's talk a little bit about that.
Right.
So yes.
Am I skipping
So-
ahead a little bit? But or we can say that we'll come to that eventually. But there is a reinforcer for parents.
Yes, there is a
There
compensation.
is compensation for participating. We know that it's difficult, and we're asking you to do something above what you would do normally. So there is a $75 compensation for your time.
It's a gift card, right? You
it's a
get a
gift- yeah.
$75
Yeah.
gift card for doing this.
Exactly.
Now, look, that's really generous, because it's not going to take them more than 10 minutes to do this on 2 different occasions, and you're going to get paid $75 for it. And then you can choose what you want to do with that $75.
Yeah.
It doesn't hurt your child, and it has the potential to change autism and the conversation around autism and how we treat autism. So I really want to encourage everybody to do this. Now, we should talk a little bit about where the collection centers are.
We do, because that is a
That's pretty important.
that is pretty important, because we've had lots of interest and queries, and there are particular clinics that are set up to receive samples. So if you live anywhere near any of these clinics, then we would welcome your participation. But it may not be possible for everyone.
Okay, so let's start with anybody who's watching who's a card family. We, of course, encourage, and I say to you, just do this. If you're a card family, you just need to do this. Don't make me come to your house and talk to you about it. I can and I will, but you need to do this. Now, I have to say that my son is 13, so I don't qualify. But if I did, I would 100% be so excited to do it, because I would be thrilled to say I was a part of this. This is a big conversation that we've been waiting for a long time. I would be so proud to say I was a part of it. So please, if you're a CARD family, first and foremost, do that. And not only, as you said before, not only are you looking for kids who are on the spectrum, if you have a family and you have a sibling who is not on the spectrum, we need that data to compare what is different between siblings.
Exactly.
Because there are a lot of things that are similar between siblings, and if we're looking at what is different between that, it gives us more data. But then you are also looking for children who are not on the spectrum, who are not related to the kiddos that are being studied, but they have to correlate in terms of age. So if you know that you've signed up a 4-year-old for the study, you're going to find a 4-year-old that is not on the spectrum, not related to that child, to be able to compare those two children as well.
Exactly.
So I know I'm going to be looking to get some friends who have neurotypical children. I say neurotypical, you know, I don't know what that means anymore, but kids who aren't on the spectrum to participate as well, because we do need some of those kids to do that. But for anybody who is not a CARD family and who says, no, I want to be a part of this, is so exciting, I want to be a part of it, you need to live within 30 miles of one of those locations that we put up, and we'll put that back up again, because we don't want your poo to defrost on the way to the center.
Exactly, that's what's critical, because we're asking you to collect the sample and then store it in your home freezer and then transport on ice to your nearest clinic. So
you got to put it on ice and get there and not stop at the grocery store. It's important because
because there are so many chemical reactions that take place, and they take place with a high velocity when it's warmer. There's more energy and they are very quick. So we like to cool the sample as much as possible, just to prevent that
stop those
to
chemical
stop it from
reactions.
potentially degrading.
Okay.
So that's why it's
And
important.
Once it goes to CARD, it stays in a freezer at CARD, and those are specific freezers. Before anybody writes to me and says they're grossed out, that's that nothing else is kept there. No. Those are under lock and key. They're research freezers. Nobody's sharing their lunch with it. Don't even write me about that. Don't go there with me. All right, and it's kept there until they have enough samples to warrant, because there's very specific packaging that it goes in to get airlifted to London.
Yes.
Where you guys receive it and you unpack it and you do what you do with it, with all those wonderful machines, and analyze that poo and pee to the end of town.
That's right.
I love that you guys get so excited about that, but that's your everyday.
Yes.
Punch in, I'm going to analyze some poo today.
That is very informative to us.
We had at least a 10-minute conversation yesterday about colour, because we found some weird and wonderful colours in one of our...
Well, anybody who has a child at some point had to go through there's a little chart where they have the poo and the different, and you get to classify your child. I guess it's just a division of that. But when it's our kids' poo, it's slightly different. But great that you guys are as excited about it as it is, because it's a study that shows great promise. Okay, and we have to have some acknowledgments for some pretty important people. I'm going to let you handle that, Torben.
Right, thanks. So yeah, we're going to thank all the people involved at Imperial College. Maybe the principal investigator, Jeremy Nicholson, of course, Professor Jeremy Nicholson, but also Professor Ian Holmes, who's the co-PI. Professor Julie Marchesi, who is responsible for the sequencing. He's doing this with Dr. Julie McDonald's, and Douglas Rees was also involved in that. And of course, we thank people at CARD, foremost Dr. Doreen, right, who's the founder of CARD, and but also Dennis Dixon, Albert Powell, Marlena Novak, Lucia Herrera, and Jonathan Narber. Also, we definitely like to thank all the funders, of course, right?
Yes.
Without them, we would not be able to do that. That is just a great project, and we are very happy that we get all that support from them.
And in particular, you mentioned before one of the funders. Let's mention them again.
That's the Christian Gerhard Jebsen Foundation.
Yeah.
So incredible that we thank people who are willing to see where there is a need, and to be willing to say, hey, I'm willing to take the time and make the investment, because that's how things get moved forward.
Yes.
I really believe that, somewhere down the line, we're going to be pointing to this study and looking at how pivotal it was in changing how we treat autism and how we diagnose autism and how we look at our kiddos with autism. And I think that that's really exciting. I'm so thrilled and honored to have had an opportunity to meet you guys and to face-to-face have this conversation. You really expanded my mind.
Thanks for having us.
Thanks for this opportunity. It's been wonderful to be able to come and to meet everyone.
Absolutely.
Here.
And I hope that as the study progresses, we can have you back on the show to talk about more information as you get data and unveil it. You know, next time you don't have to get on a plane. We can always Skype you.
Yeah,
Great.
that'd be great.
So that makes it even easier. You can do it with your fuzzy bedroom slippers on.
Yeah.
And no one will be any of the wiser. Also wanted to say, you know, we asked if people had questions. And I think looking back on it, it was kind of a crazy thing because it's like, where do you start? What's the question? Because you don't really get what's happening. But now that we've put all this out to all of you at home and you've had an opportunity to see this, you probably will have questions.
That's great.
I hope that you will want to participate. Where do they need to go? What do they need to do to sign up to do this?
So I think the first point of contact here at CARD is Lucia Herrera.
Okay.
So her email address is on the...
It's L. Herrera. L. Herrera, so that you want to spell that L.Herrera, Herrera, at centerforautism.com. So you can write to her and say, I'm interested in the study. And you know, you guys can always write to me as well if you have questions, and I will forward them to you
Right.
and your team.
Yes, we're happy to take questions as well, if that is scientific nature or not.
Okay, and so my email is S for Shannon, s.penrod, Penrod, at autism-live.com. You can always write to me and I will forward questions if you're not sure what step to take next. Fairly simple to do. Again, 18 months to 10 years old. That's the primary thing that you're trying to fill right now are kids who are on the autism spectrum. So get started. Let's get you registered and get you started. But if you have a sibling, let them know that you have a sibling. And please let other people know. You know how you've got friends who've always said, what do you need me to do for you? And there's nothing that you have that they can do, but they have a kid who's not on the spectrum. Ask them, you know what you could do for me? You can take 2 poo samples and 2 pee samples. Boy, it won't crimp their day, right? And they can do that, especially if their kiddo is the same age as your kiddo, they can do that for you and help this to move forward. That's really something that they could do for you.
It does matter, yeah.
All right. Anything that we've left out?
Oh, I hope we talked about enough about the
talked
poo.
about
Are you okay now?
I feel poo conversed. I'm yes, and I've said metabolomic. See, I got it wrong. I got it right so many times. I've said it so many times that I'm convinced I'll be able to say it again in life. I want to thank you guys, and please tell everybody at Imperial College how thrilled we are to be participating in this front row. And we look forward to everything you come back with. Go change the world.
We'll try to.
Yeah, I know.
Right,
I know
thank
you will. Thank you so much. Okay, you guys, write in with questions if you have them. That's it for our webinar today. You know, I always like to finish by saying we have a hug for you and give a hug to your kiddos. Bye-bye for now.